Factors INFIUENCINGAILEIE frequency OR deviations from Hardy-Weinberg equilibrium. The Hardy-Weinberg explanation of equilibrium in the allele frequency pattern of a population required three assumptions. 1. Individuals with each genotype must be as reproductively fit as those of any other genotype in the population. 2. The population must consist of a large number of individuals. And 3. Random mating must occur throughout the population. The Hardy-Weinberg theorem with its assumptions does not account for any change in allele frequency within populations. That is just what Hardy and Weinberg intended because their formula described the statics of a Mendelian population. Something more was required to formulate a mathematical explanation of change or dynamics in terms of allele frequencies. This need was filled by Fisher, Wright and J.B.S. Haldane, who provided additional theoretical models and superimposed the mechanisms for change in allele frequencies upon the Hardy-Weinberg equilibrium. Population statics was, thus, extended to become population dynamics. The shifts or changes in gene frequencies can be produced by a reduction in population size, selection, mutation, chance, genetic drift, meiotic drive and migration. A. Selection. 1. Evolution. Evolution has been described by Lewontin, 1967, as a process that converts variation within a population into variation between populations, both in space, race formation and speciation, and in time, the evolution of phyla. The most prominent theory regarding the driving force behind evolution is of course the Darwin-Wallace theory of natural selection proposed in 1858 by C. Darwin and A. Wallace. The natural selection theory holds that as genetic variants arise within a population, the fittest will be at a selective advantage and will be more likely to produce offspring than theorist. As the fit continue to enjoy greater survival, reproductively new species will eventually evolve. 2. Natural selection. Natural selection is generally believed to be the prominent agent for determining the relative frequency of alleles in a population. Natural selection differentiates between phenotypes in a population with respect to their ability to produce offspring. One phenotype may better survive endemic onslaughts of parasites or predators than another. One may penetrate new habitats more effectively than another. One may mate more efficiently than other. One may even prey on the other. The important point is that some natural situation or environmental feature selectively allows one organism to develop and to propagate more efficiently, and one genotype is thereby afforded greater representation in the population's gene pool. If this selective process continues over many generations, allelic frequencies will change significantly and the potential will arise for evolutionary change. Further, the natural selection process, while acting on the total phenotype, will, in fact, influence only the heritable portion of the phenotype. If a trait has a high heritability, selection can rapidly affect its frequency within a population, whereas selection will take a great deal longer to have any effect on a trait with a low heritability. Natural selection includes the following three parameters: survival rate, relative fitness and the selection coefficient. To calculate values for these parameters and, thus, best understand their meaning, we can consider the data from a particular population. For simplicity we shall consider only a single gene locus in the population defined by the A and alleles and we shall assume incomplete dominance, so that A heterozygote can be distinguished phenotypically from AA, and AA homozygotes. We then count the number of AA, and AA individuals in a given generation immediately before and immediately after some selective event, the introduction of a parasite, a change in temperature, has occurred. The representative numbers have been given in Table 7-3. From these we can calculate a survival rate, relative fitness and selection coefficient, as shown in the table. Table 7-3 relative fitness and selection coefficients calculate. SA equals 1 W equals 0.16. SA equals 1. The genotype with the largest survival rate is defined as the fittest, and it is used as the standard fourth relative fitness W of all other genotypes. Specifically, in our example, AA has a survival rate of 0.95 and S, thus, fitter than AA or AA Table 7-3. In determining values for U, therefore, all survival rates are divided by 0.95 so that the relative fitness of AA becomes 0.95, 0.95 equals 1, the relative fitness of AA becomes 0.80, 0.95 equals 0.84, and that of AA becomes 0.55, 0.95 equals 0.58. The selection coefficient S is simply 1 W. Just as W reflects the chances of an organism's reproductive success, so does S reflect the chances of its reproductive failure due to selection. They are simply two sides of the same coin. 3. Directional selection. A principal pattern followed by selection in natural populations is that of directional selection. Its effect is to eliminate or to reduce the reproductive potential of particular phenotypes in a population. Specifically, if the variance in a particular trait is normally distributed in a population before directional selection begins to operate, it might become markedly skewed in one direction afterwards. Fig. 7.2. Directional selection in its extreme form eliminates systematically the recessive homozygotes, AA, from the population. In such cases the relative fitness of AA is set at 0 and its selection coefficient, therefore, becomes 1 minus 0 equals 1. If we assume, for simplicity, that AA and AA have identical fitnesses and that no other alleles at the locus are involved, then by definition AA and AA each have fitness of 1 and selection coefficients of 0. Further, when directional selection remains in its incomplete form against homozygous recessives, then selection coefficients is less than 1 and the relative fitness W of the homozygous recessives is 1s, a number greater than 0. 4. Artificial selection. The artificial selection is simply a selection program developed by Shishi-Uma DNS rather than managed by natural situations. The process of enriching a population for a given trait normally occurs far more rapidly under the artificial situation, since all but the desired T phenotypes can be prevented from reproducing. Countless experiments have shown that it is possible to select for just about any trait present in a population, a major limitation being the heritability of the trait. Strains with particular shapes, sizes, behavior patterns, temperature optima, sexual preferences, and so on have been selected. S. Spiegelman and colleagues have even subjected isolated RNA molecules from RNA phage to artificial directional selection, and they reported that a rigorous selection for one trait can lead to the drastic exclusion of many other traits. 5. Significance of the heterozygote. When the frequency of an abnormal recessive gene becomes very low, most affected offspring will come from matings of two heterozygous carriers For example, in the human population, the vast majority of newly arising albino individuals in a XY given generation, more than 99% of them will come from normally pigmented hetfig. 7.2. The effects of directional selection on a poperozygus parents whose phenotypes are normally distributed between two extremes, X and Y. Detrimental recessive genes in a popudirectional selection acting against X and in Laschen are obviously harbored mostly in the favor of Y changes the distribution of the heterozygous state. The frequency of phenotypic variation after Goodenough and heterozygous carriers is many times greater Levine, 1974, than the frequency of homozygous individuals afflicted with the trait. Thus, an extremely rare disorder such as alkaptonuria, blackening of urine, occurs in 1 in 1 million persons. This detrimental gene, however, is carried in the hidden state by 1 out of 500 persons. There are 2,000 as many genetic carriers of alkaptonuria. For another recessive trait, cystic fibrosis, 1 out of 1,000 individuals is affected with this homozygous trait. 1 out of 16 persons is a carrier of cystic fibrosis, see Table 7-4. Genetic load, concealed variability, and price of evolution. Genetic load can be defined as the relative deficiency in viability or fecundity, or both viability and fecundity as compared to that which would occur if the population were all of the most fit phenotype. Thus, genetic load is the difference between the actual fitness and an optimum fitness, this difference being caused by the presence of suboptimal genetic variants, concealed or not, within the population. In fact, mutants arise in any species and many or most of them are deleterious. Some of these mutants may persist in the populations, i.e., one or several generations intervene between the origin and the populations of any species, including the best adapted and most successful ones, carry burdens or loads of genetic defects. One can distinguish between expressed, overt, or concealed genetic loads. 4b0y frequencies of recessive homozygotes and heterozygous carriers, source Volpe, 1980. The former comprises individuals with diseases, malformations, or constitutional weaknesses that are manifestly genetic. Concealed genetic burdens are due to recessive genes or gene complexes carried in heterozygous conditions in individuals who are themselves healthy or normal. When homozygous, however, these genes too become lethal. Types of genetic loads. According to Farnsworth, 1988, the genetic loads are of the following three types. 1. Mutational load. It refers to the deleterious effects on a population caused by recurrent mutation. 2. Segregational load. It arises when the heterozygote is the favored genotype. The formation of heterozygotes is necessarily accompanied by the generation of the less well-adapted homozygous genotypes whose lowered viability and fecundity cause the segregational load. Segregational load is, thus, the loss in average fitness of a population caused by the segregation of the less fit homozygous genotypes. Because selection acts to preserve the heterozygous genotype, neither allele, even if one orbithalethal, is eliminated from the population. As an example, in malarial regions of Africa, the loss or lowered fecundity of HBS and HBA homozygotes as compared to heterozygotes, HBA, HBS, constitute the segregational load for that locus. 3. Substitutional or transient load. It occurs when a change in the environment results in directional selection in favor of a previously neutral or unfavorable allele whose effects in the altered environment now confer greater fitness. The substitutional load, thus, consists of the relative number of deaths and lack of birth which the population must allow in order to progress from the original new state of fitness. Substitutional load is transient in that it occurs only during the replacement of one allele by another. For example, in the United States where falciparum malaria is absent, HBA homozygotes possess the best adapted phenotype, and directional selection in favor of the normal allele is occurring. As a result, the lowered fecundity and viability of HBS, HBA heterozygotes and HBS homozygotes, as compared to normal individuals, contribute a transient substitutional load to the black population of the United States. The lowered fecundity and viability of HBS, HBA heterozygotes and HBS homozygotes, as compared to normal individuals, contribute a transient substitutional load to the black population of the United States. The substitutional genetic load has been called the price of evolution, and J.B.S. Haldane, 1957, has made some theoretical calculations of the extent of this price in terms of reduced fitness or loss of individuals. He has estimated that a selection intensity of 0.1, approximately 300 generations, will be required to replace a gene at a cost in members lost by death or sterility of 30 times the average number of individuals present in a single generation. Thus, according to these calculations, approximately 10% of a population would be lost per generation if gene replacement occurred over a time span of 300 generations. Because of the enormous amount of protein, and, therefore, genetic polymorphism recently revealed at the molecular level by immunological and electrophoretic methods, these calculations pose a dilemma. Evolution requires the concurrent replacement of many genes, and to the substitutional load borne by a population must be added to the mutational load as well as segregational load contributed by stabilizing selection. Evolution requires the concurrent replacement of many genes, and to the substitutional load borne by a population must be added to the mutational load as well as segregational load contributed by stabilizing selection. It would appear that no population could withstand such a drain on its members, especially if the effects of individual subbetal genes were independent and, therefore, multiplicative. The problem, then, is how are these numerous genic polymorphisms maintained without increasing a genetic load so heavy as to cause the extinction of a population? The following two hypotheses have been proposed to resolve this problem. 1. Selection hypothesis of genetic load. This hypothesis proposes that observed polymorphisms are each due to heterozygote advantage and are maintained in a population by various types of stabilizing or balancing selection. Examples include balanced polymorphism, such as sickle cell heterozygotes, as well as cases where one or another allele or chromosomal inversion favored in different seasons, regions or stages of life cycle or in different sexes. Frequency-dependent selection also serves to maintain polymorphism, and at equilibrium the alleles involved are selectively neutral, contributing no genetic load. Supporters of the selection theory suggest that the primary cause of the extensive heterozygosity observed in nature is heterosis at hundreds or even thousands of gene loci, the cumulative effects conferring fitness. In 1967, various workers such as Sved, King, Milkman and others have proposed that the genetic load should not be estimated on the assumption that each locus contributes to this load independently and that the individual effects are multiplicative. In view of the known interaction between genes, they suggest that the values of the sums of these effects exhibit a normal distribution in a population. Since selection scans the entire organism and not single gene loci, only those individuals that fall below a certain threshold of fitness, that is, those with fewer heterotic loci than a minimum number required for average fitness, are collected from the population. As a result, members of a population are divided into two classes, those above and those below a given threshold, and under these circumstances it is estimated that perhaps 1,000 selectively maintained polymorphisms, each conferring an advantage to heterozygotes of around 1%, could persist in a population with only minimal effects as genetic load. 2. Neutralist theory of genetic load. This theory was proposed and developed by J. F. Crow, K. Kimura, T. Oda, and T. H. Jukes during 1968 and 1983. These workers suggest that the majority of thousands of genic polymorphisms discovered at the biochemical level have no significant effect on fitness and, therefore, selectively neutral, making no contribution to the genetic load. They propose that adaptation does occur through natural selection, but that selection acts primarily to remove deleterious mutations from a population. Proponents of the neutralist theory indicate that while mutations which result in the substitution in a protein of a compatible amino acid are detectable by sophisticated methodology, they may not be detected by natural selection because the function of the protein will remain essentially unchanged. Under these circumstances, the frequencies of polymorphic genes conferring no advantage or disadvantage will be determined primarily by random genetic drift rather than by selective processes. Genetic load in human populations. Estimates of the genetic load in the human population have been based principally on the incidence of defective offspring from marriages of close relatives, consanguineous marriages. It can be safely stated that every human individual contains at least one newly mutated gene. It can also be accepted that any crop of gametes contains, in addition to one or more mutations of recent origin, at least 10 mutant genes that arose in the individuals of preceding generations and which have accumulated in the population. The average person is said to harbor 4 concealed lethal genes, each of which, if homozygous, is capable of causing death between birth and maturity. The most conservative estimates place the incidence of deformities to detrimental mutant genes in the vicinity of 2 per 1,000 births. B. Mutation. Mutation is an evolutionary agent, and mutability is a required property of the genetic material if evolution is to occur. One can visualize several ways that mutation might bring about evolutionary changes. Mutation may be highly directed at a particular locus such that allele A is selectively driven to the A form. Alternatively, the mutation process might be random, but with time the A form would come to predominate over A. Finally, mutation might simply provide a population with new alleles, new mutational currency, on which any and all evolutionary agents, natural selection, for example, can act. C. Meiotic drive and migration pressure. Besides directional selection, natural and artificial, and mutation pressure, meiotic drive and migration pressure are two agents that can shift gene frequencies in a population out of a Hardy-Weinberg equilibrium. 1. Meiotic drive. The Hardy-Weinberg concept of an equilibrium population assumes the tallels will segregate in a 1 fashion at meiosis and that all gametes in the pool have an equal probability of fertilizing one another. Clearly, if gametes of genotype A have greater success in fertilization than gametes of genotype A, the frequency of A in the population should increase as A decreases. Such a mode of bringing about potential evolutionary change is in many respects a form of natural selection, but it has earned the specific designation of meiotic drive. Normal segregation ratios can be biased by a number of factors. The most extensively studied examplized segregation distorter, SD, locus in D. melanogaster. A plus SD heterozygous male transmits to his offspring many SD alleles than plus alleles. A high prevalence of SD might, therefore, be expected in natural populations, but the allele is, in fact, uncommon, its frequency being less than 0.1. Some additional selective pressures, thus, appear to counteract meiotic drive at this particular locus, presumably by agents that act against SD-carrying flies. The existence of meiotic drive is not easy to demonstrate experimentally. However, its specific role in evolution is still unassessed. 2. Migration pressure. Perhaps the most obvious way that gene frequencies in a population can be changed is the introduction of new individuals with new genotypes, a process known as migration pressure. The effect of migration pressure can be expressed mathematically as follows. In a population we can call X a particular gene is present with a frequency Q, Fig. 7.3. A certain number of individuals from this population now migrate and join population Y in which the gene in question is present at frequency QY. The immigrant, thus, comes to represent some fractionum of the total number of individuals present in the expanded version of population Y, and a new gene frequency, Q, becomes established. The value of will be equal to the contribution made by the immigrants Q, M plus the contribution made by the original population Q, 1M, where 1 meter represents the proportion of non-migrants. Therefore, QQ equals Q, MTQ, 1M. And the change in Q after one generation in such a population becomes AQ equals QQ equals G, MG, plus MQG, Q equals MQ, Q. When numerical values are substituted into this equation, it becomes clear that significant changes in the value of Q can result if these two populations differ only slightly in the frequency of a given gene and if only a moderate degree of migration occurs between them. D. Random genetic drift. Random fluctuation in allele frequencies, called genetic drift, also occurs in breeding populations. The effect of genetic drift is negligible in large populations, but in small breeding populations all the limited number of The effect of genetic drift is negligible in large populations, but in small breeding populations all the limited number of progeny might be of the same type with respect to certain gene pairs because of chance alone. Should this happen, fixation or homozygosity will have occurred at the locus concerned. Fixation is defined as gene frequency reaching P equals 1.00 or Q equals 1.00. Chance fluctuations may or may not lead to fixation. BBOBLLI. B. However, in Asia, the ancestral population. Accordingly, the possibility. Evolutionary geneticists interpret this. The Y population of mobs receives some immigrants from the X population, an event that changes its frequency of the white allele after Goodenough and Levine, 1974. E. Founder principle. When a few individuals or a small group migrate from a main population, only a limited portion of the parental gene pool is carried away. In the small migrant group, some genes may be absent or occur in such low frequency that they may be easily lost. The unique frequency of genes that arise in population derived from small bands of colonizers or founders has been called the founder effect or founder principle. This principle was proposed in 1956 by Harvard evolutionist Ernst Mayr. The founder principle essentially emphasizes the conditions or circumstances that support the operation of Sewall-Wright's genetic drift. For example, North American Indian tribes, for the most part, lack the gene IB that governs type B blood. However, in Asia, the ancestral home of the American Indians, the IB gene is widespread. The ancestral population of Mongoloids that migrated across the Bering Strait to North America might have been very small. Accordingly, the possibility exists that none of the prehistoric immigrants happened to be blood group B. It is also likely that a few individuals of the migrant band did carry the I gene but they failed to leave descendants. Evolutionary geneticists interpret this peculiar feature in terms of genetic drift. Most of the North American Indians possess only blood group O, or stated another way, contain only the blood allele I. With few exceptions, the North American Indian tribes have lost not only blood group allele I but also the allele that controls type of blood I. The loss of both alleles, I and IB, by sheer chance perhaps defies credibility. Indeed, many modern students of evolution are convinced that some strong selective force led to the rapid elimination of the and I genes in the American Indian populations. If this is true, it would provide an impressive example of the action of natural selection in modifying the frequencies of the genes in a population. Genetic polymorphism. One of the most characteristic features of any natural population is its diversity. This diversity is obvious when we consider the human species, for we are attuned to sensing differences in human appearance, personality, sexuality, and so on. In the populations of flies or dandelions such well-marked diversity does not occur, but it exists nevertheless. In genetic terminology, natural populations are said to be polymorphic. Polymorphism is most apparent when it affects a visible or behavioral phenotype, but is not at all restricted to such traits. R. Lewontin and J. Hovey, in 1966, undertook the first extensive analysis of protein polymorphisms in natural population of Drosophila pseudoobscura by subjecting extracts of individual flies to get electrophoresis and observing the rates of migration of various proteins, which represented 18 gene loci. They found, quite unexpectedly, that many of the proteins existed in the population in the form of isoelectric variants, meaning that for a given type of protein some individuals possessed a fast-migrating species and others a slow-migrating species. Numerous subsequent studies of such diverse species as barley, wild oat, horseshoe crab, mouse and man have all produced the same result. An abundance of protein polymorphism is found wherever it is sought. Protein polymorphism signals the existence of allylism, and it has been estimated that 20 to 50% of all structural gene loci in a given species exist in 2 or more allelic forms in any given population. The polymorphism may arise in a population by the following 3 basic avenues: transient polymorphism, balanced polymorphism and random fixations of natural mutations. 1. Transient polymorphism. Transient polymorphism is a byproduct of directional natural selection. If we imagine Theta underscore has aselective advantage of era, then with time A should proceed toward fixation at EP equals 1, and A should proceed toward elimination ATQ equals 0. While this process is occurring, both A and A will be present in the gene pool and A heterozygotes will be present in the population. As name implies, transient polymorphism represents a temporary situation. For example, during the course of industrial melanization, both dark and light mobs would be expected to cohabit the Manchester trees for the interim, but the proportion of light mobs would be seen to diminish with time as dark mobs gradually predominated. 2. Balanced polymorphism. Balanced polymorphism is also a relatively permanent kind of equilibrium in which alleles A and A are present in the population at some steady state frequencies. Balanced polymorphism is originated by disruptive or diversifying selection and heterosis. 3. Random fixation of natural fixation. The random fixation of natural fixation method of origin of polymorphism is also called neutral mutation, random genetic drift hypothesis or non-Darwinian evolution, and this idea has been developed by S. Wright and Kimura. This hypothesis is based on the following 2 assumptions. 1. The first assumption states that selectively neutral mutations can occur in genes that code for proteins. This will clearly be true in the case of synonymous mutations in which one codon is replaced by another codon dictating the same amino acids, but it is also proposed to be true in the case of mutations that lead to amino acid substitutions. The idea is that an acidic amino acid might occur in an unimportant region of the protein, with the result that the emergent mutant protein is identical to the original in all functional aspects. The second assumption states that neutral alleles, being neither selectively advantageous nor disadvantageous, simply drift in the gene pool. Thus, if a neutral mutation arises in woman's germ cell and this germ cell gives rise to a female child, the probability is about 0.5 that the mutant allele will be transmitted to a grandchild and 0.5 that it will not. If it is not, then G becomes equal to 0 and the allele is lost. Polymorphism of cytochrome C of all eukaryotes and hemoglobin protein of all vertebrates strongly support the Wright-Kimura hypothesis. POPUIATION Genetics and evolution. Thus, population genetics has provided great support to the idea of organic evolution. Various aspects of evolution can be reviewed in terms of population genetics as follows: speciation, original population. The process of formation of event cause MG. LO. Bottleneck new species is called speciation. 2 genetically divergent populations can form new species only when they become geographically isolated from each other. If a large I population is fragmented into 2 copyright time T or more units which are geographically isolated from one another, each independent unit follows different evolutionary paths for the following reasons. 1. Each isolated unit of a population may have its own type of mutation which provides raw materials for organic diversity. 2. The mutations and gene combinations which appear in different isolated population units will have different adaptive values in the new environments. 3. The organisms which originally colonize a certain geographical area and form an isolated population may not be representative of the group from which they came, so that different gene frequency resulting population. In a population bottleneck, a population undergoes a drastic reduction in size as a result of a natural catastrophe or overhunting. Then only a few individuals are left to contribute genes to entire future population. The cheetah passed through a population bottleneck in the recent past, resulting in an almost loss of genetic diversity. Size exists from the beginning. 4. The size of the new population may become quite small at various times so that a genetic bottleneck is formed, from which all subsequent organisms will arise. 5. During the period of small population size, the gene frequencies will fluctuate in unpredictable directions. The fluctuation in gene frequency is called genetic drift. RUAVNIARO. Genetic changes in a species helps it to adjust to new or altered environmental conditions. Evolution above species level. Adaptation, adaptive radiation, microevolution, macroevolution, megaevolution, punctuated equilibria and related phenomena. Adaptation can be defined as occurrence of genetic changes in a population or species as a result of natural selection so that it adjusts to new or altered environmental conditions. Thus, any characteristic that is advantageous to a particular organism or population is called an adaptation. Savage, 1969. For example, the lungs of land vertebrates make possible gaseous exchange between these organisms and the air and form an adaptation for terrestrial life. The fact of adaptation is confirmed by the diversity of life and the thousands of environmental situations inhabited by living organisms. For various examples of adaptation, see Part 5 Ecology, Chapters 3 and 18. Adaptive radiation. The term adaptive radiation has been coined by H. F. Osborn, 1902, for explaining evolution, from a single ancestor, of a number of descendants with a great variety of adaptations to different niches. More aptly stated, the phenomenon of adaptive radiation is the diversification of a dominant evolutionary group into a large number of subsidiary types adapted to more restrictive modes of life, different adaptive zones, within the range of the larger group. According to George Gaylord Simpson, 1940, 1953, adaptive radiation is the rapid proliferation of new taxa, species, from a single ancestral group. Certain authors of evolution biology such as Savage, 1969, Stanley, 1979, and Volpe, 1985, have used an entirely new term macroevolution for Osborniswaf of adaptive radiation. Examples of adaptive radiation. 1. Adaptive radiation in Darwin's finches. A classical example of speciation involving the interplay of complex forces, such as isolation, competition, adaptation, etc., leading to adaptive radiation at the species level is provided by Darwin's finches. These birds belong to largest family of birds, the Fringillidae, and live in Islas Encantadas, Galapagos Islands, in the Pacific Ocean. Significantly, the study of these small dark birds in their native habitat gave Darwin his first insight into evolutionary processes. David Lack, 1947, 1953, 1969, has analyzed the phenomenon of adaptive radiation of Darwin's finches very carefully. The Galapagos Islands lie on the equator about 600 miles west of South America, i.e., Ecuador. In Archipelago, there are 5 large islands in the group, with 19 smaller ones and 47 rocks. These islands are of volcanic origin, figure 8.1. They were never connected with the mainland of South America. The rugged shoreline cliffs are of gray lava and the coastal lowlands are parched, covered with cacti and thorn bushes. In the humid uplands, trees flourish in rich black soil. The present-day assemblages of Darwin's finches descended from small sparrow-like birds that once inhabited the mainland of South America. The ancestors of Darwin's finches were early migrants to the Galapagos Islands and probably the first land birds to reach the islands. These early colonists have given rise to 14 distinct species, each well adapted to a specific niche. 13 of these species occur in the Galapagos, one is found in the small isolated Cocos Island, northeast of Galapagos. Not all 13 species are found on each island. These 14 species belong to 4 genera: 1. Geospiza, 2. Camarhensis, 3. Certhidia, and 4. Phaneroloxias. The most conspicuous difference among the species are in the sizes and shapes of the beak, which are correlated with marked differences in feeding habits. 6 of the species are ground finches, with heavy beaks specialized for crushing of seeds. Some of the ground finches live mainly on a diet of seeds found on the ground. For example, there are 3 seed-eater ground finches: small-beaked Geospiza fuliginosa, medium-beaked Geospiza fortis, and large-beaked Geospiza magnirostris, occur together in the coastal lowlands of several islands. Each species, however, is specialized in feeding on a seed of a certain size. The small-beaked finch feeds on small grass seeds, whereas large-beaked finch eats large, hard fruit. Other ground finches feed primarily on the flowers of prickly pear cacti. The cactus eaters possess decurved, flower-probing beaks. Their beaks are thicker than those of typical flower-eating birds. All other species are tree finches, the majority of which feed on insects in the moist forests. One of the most remarkable of these tree dwellers is the woodpecker finch, figure 8.2. It possesses a stout, straight beak, but lacks the long tongue characteristic of the woodpecker. Like a woodpecker, it bores into wood in search of insect larvae, but then it uses a cactus spine or twig to probe out its insect prey from the excavated crevice. Equally extraordinary is the warbler finch, which resembles in form and habit the true warbler. Its slender, warbler-like beak is adapted for picking small insects off bushes. Occasionally, like a warbler, it can capture an insect in flight. Thus, the original ancestral stock of finches on the Galapagos diverged along several different paths. The pattern of divergence is reflected in the biologist's scheme of classification of these birds. All the finches are related to one another, but the various species of ground finches evidently are more related by descent to one another than to the members of the tree finch assemblage. As a measure of evolutionary affinities, the ground finches are grouped together in one genus, Geospiza, and the tree finches are clustered in another genus, Camarhensis. The different lineages of finches have been portrayed in figure 8.3. Darwin's finches-like adaptive radiations within ecologic islands of any sort are called coincideradiations, Stanley, 1969. Darwin's finches provide circumstantial evidence for the origin of a new species by equals means of geographical isolation. 2. Adaptive radiation in penguins. G. G. Simpson, 1953, has cited evidence that when a group of organisms enters a neoadaptive zone previously unoccupied by the group, there may be rapid bursts of speciation and adaptive divergence into a variety of ecological niches. In Fig. 8.4, we see Simpson's visualization of such adaptive radiation for penguins. Note that there are 3 zones for penguins: 1. Aerial flight, 2. Aerial and submarine flight, and 3. Submarine flight. These zones were sequentially invaded by penguins in their evolution, and they are now extinct in the aerial flight zone or subzone. 3. Adaptive radiation in reptiles. The class Reptilia first appeared in the fossil record in Pennsylvanian times, 250 million years ago. The ancestral reptilian stock initiated one of the most spectacular adapts. Cocos Island, Galapagos Islands at Culpeper B. Wenman. Type radiations in life's history. Adaptive Abinger #I equals tower radiation of the reptiles occurred between Bindle Permian and Cretaceous times, and living. 4 reptiles are derived from Cretaceous ancestors. V. James. Tors. The Mesozoic era, during which the Geoffiorum reptiles thrived, is often referred to as the indefatigable age of reptiles. OLA 1. Albemarle Barrington Chatham I. Charles Mag. Suffering extinction. Dinosaurs were remarkably diverse. They varied in size, bodily form and habits. Some of the dinosaurs were carnivorous, such as the gigantic Tyrannosaurus, whereas others were vegetarians, such as the feeble-toothed but elephantine Brontosaurus. The bulky body of Brontosaurus weighed 30 tons and measured nearly 70 feet in length. Not all dinosaurs were huge, some were no bigger than chickens. Some dinosaurs strode on 2 feet, others had reverted to 4. The exceedingly long necks of certain dinosaurs were adaptations for feeding on the foliage of tall coniferous trees. Geospiza. Ground finches. T1 foot 5 Fuliginosa, trademark Certhidia. U. Small, Lee. Warbler finches. Fortis conorostris. Medium. E. I_1. H pallidus F. S Euro andans equals Polis votia. Exclamation mark. Setacula, parvolisk. Camaranchus, tree finches. Magnalrostrals difficilis. Large. Paneroloxias. Coco SFL finches. I. Anornata. Crassirostris ancestral stock. Figure 8.3. Evolutionary tree of Darwin's finches based on Lack's findings. Darwin's finches evolved from a common stock and are represented today by 14 species. The species are assembled in 4 genera, after Volpe, 1985. The dinosaurs were descended from the Thakodonts, slender, fast-running lizard-like creatures. The Thakodonts gave rise also to bizarre reptiles that took to the air, the pterosaurs. These, dragons of the air, possessed highly expansive wings and disproportionately short bodies. The winged pterosaurs extincted before the end of Mesozoic era. Another independent branch of the Thakodonts led to exceptionally more successful flyers, the birds. The origin of birds from reptiles is revealed by the celebrated Archaeopteryx, a Jurassic form. The feathered creatures possessed a slender lizard-like tail and a scaly head equipped with reptilian teeth. Certames Ozoacreptiles returned to water. The streamlined, dolphin-like ichthyosaurs and long-necked, short-bodied plesiosaurs were marine, fish-eating reptiles. The ichthyosaurs were expert swimmers. Their limbs were fin-like and their tails were forked. The plesiosaurs were efficient predators, capable of swinging their heads 40 feet from side to side and seizing fish in their long, sharp teeth. These aquatic reptiles breathed by means of lungs. They did not redevelop the gills of their very distant fish ancestors. Indeed, it is axiomatic that a structure once lost in the long course of evolution cannot be regained. This is the doctrine of irreversibility of evolution, or Dallow's law, after Eloise Dallow, 1895, the eminent Belgian paleontologist to whom the principle is ascribed. Further, among the early reptiles present before Mesozoic days, i.e., Permian of Paleozoic era, were the Polychosaurs, e.g., Dimetrodon, notable for their peculiar sail-like extensions of the back. The function of the gaudy sail is unknown, but it should not be thought that this structural feature was merely ornamental or useless. It appears that the sail of Polychosaurs was a functional device to achieve some degree of heat regulation. Polychosaurs gave rise to an important group of reptiles, the therapsids. These mammal-like forms bridge the structural gap between the reptiles and the mammals. The history of the reptiles attests to the ultimate aerial flight. Fate to many groups of organisms, extinction. The reptilian dynasty collapsed before the close of the Mesozoic era. Of the vast host of Mesozoic reptiles, relatively few have survived to modern times. The ones that have include the lizards, snakes, crocodiles and turtles. Terrestrial. RECNT. Some authors maintain that the active Thakodonts and dinosaurs were already warm-blooded, endothermic, rather than, as often assumed, cold-blooded, ectothermic. An endothermic animal is not dependent on external sources of heat, but has the capacity to produce and control its own internal heat. The long reign of the dinosaurs could reflect, in MIOCNE. Figure 8.4. Adaptive Radiation Evolution of Penguin after Hamilton, 1967. Aerial and submarine flight. Penguins. Part. The capacity for endothermy. 4. Adaptive radiation in eutherian mammals. The eutherian placental mammals provide another classical example of the process of adaptive radiation. In this case, from a primitive, insect-eating, insectivorous, short-legged, rat-like terrestrial Creature that walked with the soles of its feet flat on the ground have evolved all the present day. Types of mammals. Thus, in respect to limb structure, among eutherian mammals, adaptive radiation occurred in the following 5 different lines. I. Arboreal. Own evolutionary line radiates to form arboreal forms which have adapted limbs. For life entries, e.g., squirrels, sloth, monkeys, etc. The legs which are adapted for climbing are called scansorial. E. Volant or aerial. Another line leads to arboreal forms such as monkeys have adapted limbs for life in trees. Aerial or volant, representing mammals adapted for flight, e.g., bats. Somewhere along this line we can place the gliding mammals such as flying squirrel. The arboreal and aerial forms not arose independently from the terrestrial forms, but perhaps through semi-arboreal or climbing ancestor. Fig. 8.6. E. Cursorial. Third line of radiation gives rise to cursorial forms such as horses and antelopes. They have developed limbs suitable to rapid movement. The cursorial mammals have the following 3 types of adaptation in their foot postures, such as 1. Plantigrade, i.e., walking with whole sole of the foot touching the ground, e.g., bears and primates including human beings. 2. Digitigrade, i.e., digits touch the ground and are provided with pads on their ventral side which absorb the shock and help in making stealthy approach towards the prey, e.g., lion, tiger, leopard, cat, wolf and dog. And 3. Unguligrade, i.e., animals walk and run on the tips of their fingers and toes which are shielded by hoofs. E.g., artiodactyls are double-hoofed animals such as cattle, sheep and buffaloes, and perissodactyls are single-hoofed animals such as horses, asses and zebras. IV. Fossorial. Fourth line of radiation formed the burrowing mammals, the fossorial mammals. Some of the fossorial mammals, such as moles, have modified their forelimbs for digging but they are poorly adapted for locomotion on the ground, while other fossorials such as pocket gophers and badgers are expert diggers but they can also move readily on the surface of ground. V. Aquatic. Fifth line of radiation leads to the aquatic mammals. Whales and porpoises. Having limbs strongly adapted for aquatic life. But they cannot move about on land. While seals, sea lions and walruses have also strongly modified limbs for aquatic life but they are also able to move about on land. The third group includes accomplished swimmers such as otters and polar bears which are equally at home in water or on land. Lastly, animals' legs modified for walking are called ambulatory and those that are adapted for jumping are called saltatory. 2. The Kogients. NII. Among eutherial mammals, adaptive radiation is also applied to teeth, or mode of feeding. Fig. 8.7. 5. Adaptive radiation in the marsupial metatherian mammals. A remarkable example of adaptive radiation in animals is the tremendous diversity of marsupials in Australia. In many ways they parallel the adaptations of placental mammals in the rest of the world, affording striking examples of parallel evolution. Thus, in the absence of placental mammals, marsupials developed grazing forms, some kangaroos burrowing forms, marsupial moles forms resembling tree and flying squirrels, flying phalangers, pygmy gliders forms resembling arboreal eutherians, teddy bear or koala and tree kangaroo forms containing rodent-like dentition wombats, marsupial mouse rabbit-like forms, hare wallabies, wolf-like carnivores, Tasmania wolves, badger-like carnivores, Tasmania devil, and anteating carnivores, banded anteater. Figure 8.8. Simpson's adaptive grid and macroevolution. G.G. Simpson, 1953, has developed a handy conceptual framework for describing major evolutionary patterns. We already know that at any moment in time the interaction of organisms and arboreal, aerial or volant, living chiefly flying animals, their environment define a series of broad or narrow adaptive fields or zones. All members of the major group, for example the crustaceans, share one major adaptive zone because of their common possession of a complex general adaptations. Within the broad zone, each species of crustacean occupies a distinct but narrow field of its own because of the species' peculiar combination of special and general adaptations. Each kind of organism is an adaptive type discontinuous from other adaptive types. For example, a crab is distinct from a barnacle, both are crustaceans, a snake from a man or a sunflower. Simpson has suggested that for purposes of discussion the adaptive types or zones may be represented diagrammatically as bands or pathways on an adaptive grid. Figure 8.9. The discontinuities between the major zones A, B, C, D are ecologic discontinuities or unstable ecologic zones. The adaptive zone itself is an ecologic role or characteristic relationship between organism and environment. Although the actual grid is of course very complex, comprising many subzones, 1, 2, 3, a simplified form is satisfactory for our discussion. The diagram is extremely useful in attempting to describe adaptive evolutionary change. The evolution of adaptation may be summarized in terms of grid as follows. Changes in adaptation involve movement of evolutionary lines within subdivisions of the subzones. Microevolution, either from one major zone or subzone into others. Macroevolution, or from one major set of zones into another. Megaevolution. The basic feature of evolution above the species level is the movement of a group of organisms into a new adaptive zone. In order to move across the zone of ecologic instability, i.e., an environment of strong negative selection, into the new adaptive zone, an organism must have evolutionary and ecologic access to it. In other words, the figure 8.9. Simpson's adaptive grid. Major adaptive zones or intergroup must already have some charted by letters and subzones by Arabic numbers, ecology. Characteristics adaptive to the new Kali unstable zones are shaded, after Savage, 1969. Zone, and the zone must be unoccupied by a strong competitor. An example is provided by the evolution of terrestrial plants, figure 8.10. T.I.M.E. A good example of adaptive grid is provided by the evolution of lungless salamanders. Evolution of lungless salamanders. Plethodontidae family of salamanders, amphibians, is the most successful, having numerous groups of living salamanders found in Europe, North and tropical America. The adult of the ancestral stock probably occurred in and around the margins of small mountain streams. They apparently laid delicate eggs in the water which hatched into a free-living aquatic larva with functional gills. After undergoing further development, the larva changed into semi-aquatic salamander. A number of living genera of salamanders such as Desmognathus, Eurysea, Gyrenophyllus, Pseudotriton and Typhlotriton, retain these habits and continue to occupy the ancestral semi-aquatic adaptive subzone. From this, several descendant evolutionary lines have become completely aquatic in habits. In one genus, Lorognathus, which is closely allied to Desmognathus in the semi-aquatic group, the adult is strictly aquatic, hides under rocks on the bottom of fast-moving mountain streams. A second group of completely aquatic forms never transform, but become sexually mature and breed while still in a gilled larval state. Some species of Eurysea and Gyrenophyllus have evolved by this means, while two distinctive genera allied to Eurysalsalib in paleogenetic subzone, i.e., one arval, have developed sex organs and behave like the adults. Figure 8.11. One species of Desmognathus and the genus Hemidactylium occupy the semi-terrestrial subzone. The eggs of these salamanders are unspecialized aquatic types which are deposited in moist places on land. When the larvae hatch, they are washed into streams where they undergo further development. Marine. Some Plethodontidae have also invaded a second major adaptive zone, the terrestrial zone. Eggs of lungless salamanders in this zone have a special protective capsule and are deposited on land. Development proceeds directly and salamander hatches out of the egg. No free-living larval stage occurs in the life history of these forms. These salamanders have invaded the terrestrial zone 3 times. The earliest of these invasions has resulted in adaptive radiation into 12 genera. The terrestrial forms or their descendants later occupied the semifossorial, burrowing, and arboreal, free-dwelling subzones. At much late date, one genus, Phaeognathus, and a species of a related genus, Desmognathus, each seem to have independently penetrated the terrestrial adaptive zone. These forms also possess capsulated eggs with direct development. Thus, when a new major zone or group of zones is occupied, the first zone entered is usually the widest and requires the least special adaptation. Later, more special zones will be occupied. The initial breakthrough to the new zone is due to general adaptation. Subsequent evolution leads to specialization. This pattern also fits the macroevolutionary speciation, adaptive radiation, of Darwin's finches. Mivarts Dayema and Preadaptation. St. Geogray Jackson Mivart, 1871, wrote in his book, On the Genesis of Species, that he could not see how evolution would proceed by the gradual accumulation of small changes, since most intermediate character states would be maladaptive. For example, what use is a partially formed winged torreptile? He, therefore, suggested that evolution would proceed in jumps, and that basis of these was the acquisition of useful characters. Mivart's dilemma has been explained by invoking preadaptation. The intermediate structures act in a peculiar way for which they are well suited, but by chance they are appropriate for other roles which they are able to play after further elaboration. For example, a flap of skin between the forelimb and the trunk might have evolved in reptiles as a thermoregulatory device and then have been found of use in gliding away from enemies, ultimately giving rise to a wing. Similarly, the air bladder of fishes was not evolved so that they might one day invade land but as an accessory respiratory organ for use in the aquatic environment. However, once evolved, air bladders were of use as lungs when new environmental problems, such as the drying of ponds, appeared. See Callow, 1983. Microevolution, macroevolution, MEGAEVOLUTION and hypothesis of punctuated equilibria. Goldschmidt 1940 divided evolution into microevolution, that of subspecies, and macroevolution, that of species and genera and perhaps also of higher categories. Simpson, 1953, has proposed the additional term, megaevolution, for really large-scale evolution, such as that of families, orders, classes and phyla. Evolution at these levels receives much attention from students of the fossil record. B. Wrench, 1959, has modified the terms microevolution into infraspecific evolution and macroevolution into transspecific evolution. Callow, 1983, has regarded macroevolution and cladogenesis as synonyms. For several years, 1976 to 1982, Stephen Jay Gould and Niles Eldridge of the American Museum of Natural History have questioned the conventional view that evolutionary changes in the distant past are principally the outcome of the gradual accumulation of slight inherited variations. They advocated that most evolutionary changes have consisted of rapid bursts of speciation alternating with long periods in which the individual species remain virtually unmodified. Gould and Eldridge maintain that most lineages display such limited morphological changes for long intervals of geologic time as to remain in stasis, or in equilibria. Conspicuous or prominent evolutionary changes are concentrated in those brief periods, punctuations, when the lineages actually split or branch. This is called hypothesis of punctuated equilibria. Further, branching from a single lineage, or a single line of descent, during macroevolution, will ultimately produce a cluster of lineages known as clade. Thus, branching sometimes is called cladogenesis. When referring to a clade or to a group of related clades, it is common to use the adjective. Genetic changes in a species helps it to adjust to new or altered environmental conditions. Evolution above species level. Adaptation, adaptive radiation, microevolution, macroevolution, megaevolution.